Okay, imagine for a second you've got a pair of molecular scissors, and these scissors can find any specific sentence in a 3 billion letter book, cut it right out, and just let the cell patch it up. For a long time, that is exactly how we thought about gene editing with CRISPR-Cas9. But what if you don't want to clumsily cut the page? What if instead you just wanted a pencil eraser, or honestly even better, a molecular word processor that could find a typo and seamlessly type right over it? Well, welcome to the Explainer. Today, we're moving way beyond the molecular scissors of standard CRISPR and diving into the absolute cutting edge of genetic engineering, CRISPR 2.0 and 3.0.
So here's our roadmap for today's Explainer. First, we'll look at the problem with double-strand breaks. Second, base editing and chemical conversion. Third, evolving an entirely new enzyme. And finally, prime editing, the ultimate search and paste tool. Let's get right into it.
Section 1. The problem with DSBs, the flaw of the scissors. Let's start by looking at the fatal flaw of traditional CRISPR-Cas9 and why we so desperately needed an upgrade in the first place.
Now, standard CRISPR-Cas9 is incredibly powerful, no doubt about it, but it's got one fundamental limitation. It creates a double-strand break, or DSB. It literally chops right through both strands of the DNA helix. And when that happens, the cell absolutely panics. It triggers this default repair pathway called non-homologous end joining, or NEET, to just furiously glue those broken ends back together. The cell's priority here is speed, not accuracy. So this process is inherently messy and error-prone, often introducing completely random insertions or deletions. Now look, if your goal is to deliberately disrupt or just knock out a gene, fantastic. But if your goal is to carefully and safely correct a delicate genetic sequence, well, breaking the DNA spine and just hoping for the best is a terrible strategy. And that is exactly why the field shifted toward precision editing, which relies on chemical modifications and single-strand nicks for super predictable, highly controlled repair.
And here is why this is absolutely crucial. A landmark 2019 analysis found that roughly 58% of known pathogenic human genetic variants are single nucleotide changes. Think about that. That is literally one wrong letter out of 3 billion. So for more than half of all genetic diseases, we don't need to knock out genes or make these massive insertions. We literally just need to change one single letter to another. We need an eraser, not a pair of crude scissors.
Section 2. Base editing. Chemical conversion without a DSB. This brings us to a massive paradigm shift that came out of David Liu's lab in 2016, and it represents a huge leap in how we interact with the genome.
The major conceptual advance here is that instead of cutting both DNA strands, base editors directly and chemically convert one DNA base into another. How do they do it? They take a modified Cas9, one that only nicks the DNA instead of fully severing it, and fuse it to a DNA-modifying enzyme called a deaminase. So you've got two main families here. First is the Cytosine Base Editor, or CBE, which chemically converts a cytosine to a uracil, which the cell ultimately just reads as a thiamine, so C becomes T. Then you've got the Adenine Base Editor, or ABE, which chemically converts adenine to inosine, which the cell reads as guanine, so A becomes G.
Let's actually walk through how the Cytosine Base Editor pulls this off step by step, because it's pretty wild. First, the guide RNA directs the whole complex to the target. It binds to it, peeling apart the DNA helix to form what we call an R loop. This displaces the non-target strand, leaving it temporarily single-stranded. And this is a huge detail, because that attached deaminase enzyme, it can only act on single-stranded DNA. So, while the DNA is unzipped, the deaminase scans a really specific editing window, usually just positions 4 through 8 of the sequence, and chemically deaminates the cytosine into uracil. Finally, and this is the clever part, the Cas9 nicks the other strand, the unedited one. This basically tricks the cell's repair machinery into using our newly edited strand as the template during repair. So it permanently locks in the change, and after replication, boom, we have a pristine T exactly where we wanted it. And the best part? 0 double-strand breaks required.
Section 3. Evolving a new enzyme. The ABE story. So let's see how the Adenine Base Editor actually builds on this concept, because frankly, creating the ABE required a leap of bioengineering that sounds straight out of a sci-fi movie. You see, making a cytosine base editor was conceptually straightforward, because enzymes that convert C to U on DNA already existed out in nature. But for adenine? Nope. No natural enzyme was known to deaminate adenine in DNA. A to G editing simply didn't exist in the natural world. So David Liu's lab just forced it into existence. They started with this bacterial enzyme called EEC-TAD-A, which normally only works on RNA, and they put it through 7 grueling rounds of directed evolution in the lab. They mutated it, selected the best performers, mutated those again, until they finally produced TAD-A star. This is an entirely unnatural, lab-made mutant enzyme that works perfectly on DNA. Truly incredible stuff. Now, you might be wondering, why go through all that insane effort? Well, to put it into perspective, this specific A to G conversion addresses approximately 48% of known pathogenic point mutations in the human genome. Nearly half. Just think about that. Almost half of all disease-causing single-letter typos could potentially be fixed by this one artificially evolved enzyme. So, just to quickly recap our two distinct molecular pencils here. The CBE uses naturally derived apobec enzymes to convert C to T, going through a uracil intermediate. The ABE, on the other hand, uses that laboratory-evolved Theta enzyme to convert A to G, going through an inosine intermediate. But they both use a nickase Cas9 to gently bias the cellular repair, and crucially, neither of them requires a messy double-strand break. They are just incredibly elegant tools.
Section 4. Prime editing. Search and paste. Now, base editors are absolutely brilliant, but they do have limitations. They can only make specific transition mutations, and only if that target letter happens to fall perfectly within that tiny editing window. So, what if you want to make a different type of mutation, or insert a missing sequence? Well, that brings us to our final upgrade, prime editing. Published back in 2019, prime editing is, for all intents and purposes, a true search and replace function for the genome. And the absolute genius of this whole system lies in its guide RNA, which is called a pegRNA. Instead of just acting as a simple GPS to find the target, the pegRNA is actually extended at the end. It carries a primer binding site to anchor the DNA and an RT template. And get this, the RT template is the actual text you want to paste into the genome. The guide RNA itself is literally carrying the blueprint for the edit.
Let's look at the choreography of how this actually plays out. Step 1. The prime editor complex binds the target and nicks just one strand of the DNA, creating this loose little flap. Step 2. The primer binding site on our special pegRNA reaches over and grabs hold of that loose DNA flap. Step 3, and here is the absolute magic. The Cas9 is fused to a reverse transcriptase enzyme. This enzyme literally reads the RNA blueprint we attach, and it copies that new edited sequence directly onto the DNA flap. Step 4. The cell naturally resolves this flap, safely clipping off the old unedited DNA. And finally, Step 5. The cell permanently seals that newly written sequence right into the genome. It searches, it copies, and it pastes. I mean, the capabilities of prime editing are just staggering. Because you're directly writing new text, you aren't limited to just C to T or A to G anymore. Prime editing can theoretically perform all 12 possible point mutations. It can paste in small insertions, it can orchestrate small deletions, and it does the whole shebang without ever creating a double-strand break, and without needing some external donor DNA template floating around. It is a completely self-contained genetic word processor. We started with molecular scissors that uncontrollably shattered DNA to break genes. Then we progressed to base editors that could chemically erase and rewrite very specific single letters. And now, we have prime editors that can search and paste entirely new sentences directly into the code of life itself. The tools are here. They're ready. So the only question left is, with the ability to search and replace literally any letter in our DNA, what typos will humanity erase first? Thank you so much for watching this explainer. And as always, keep learning.